In a fraction of a second, a viper strike delivers a complex biochemical payload. This venom acts as a high-velocity weapon, injected with mechanical precision directly into the target. Looking at a cross-section of the viper's skull reveals the mechanics of this delivery system. A movable maxillary bone acts as a hinge, pushing against the prefrontal bone to snap the fangs from a folded resting position out to exactly 90 degrees. Those fangs operate as true hypodermic needles. The groove along the tooth is completely enclosed, creating a sealed, pressurized tube. Driving that pressure is a muscular pump. The masseter and temporal muscles forcefully contract around the venom gland, pushing the liquid down through a duct and directly out the distal orifice of the fang. This folding hinge is highly specialized to vipers. Elapids, a family that includes cobras and mambas, rely on a different anatomical model featuring short, permanently fixed tubular fangs at the front of the mouth.
Colubrid snakes utilize yet another structure, grooved, rear-facing fangs situated at the back of the jaw, requiring them to chew the venom into their target rather than injecting it through a sealed tube. Regardless of the specific bone structure, the physiological goal remains identical. The entire cranial apparatus is designed to act as a high-pressure biological syringe capable of breaching a prey's dermal armor. Tracing the plumbing backward from the fangs leads directly to the biological factory producing the payload, the venom gland, located just behind the eye. Anatomically, this structure is a highly modified parotid salivary gland. This places the origin of the venom apparatus firmly within the snake's digestive system. Inside the gland, the liquid venom is held in large storage sacs called alveoli, waiting in reserve for deployment. The substance stored in these sacs is incredibly dense. If you remove the water content, 90 to 95% of the venom's dry weight consists entirely of active proteins and polypeptides. By modifying a standard salivary gland, the snake weaponizes its digestive processes. It synthesizes a concentrated biochemical matrix, instantly overwhelming a target's physiological defenses. Once injected, the payload enters the bloodstream. For vipers, the primary agents doing the damage are hemotoxic and cytotoxic proteins. This diagram isolates a red blood cell membrane. One specific enzyme, phospholipase A2, attaches directly to the phospholipid bilayer. Upon attachment, it transforms the phospholipid into a lysophospholipid, essentially turning it into a soap. This rapidly compromises the membrane, violently rupturing the cell through hemolysis. Simultaneously, a second class of hemotoxins called metalloproteinases goes to work. Metalloproteinases, such as mucrolysin, specifically attack and degrade the vascular endothelium, the delicate tissue lining the inside of the blood vessels. This degradation leads to rapid, severe internal hemorrhaging. These hemotoxic agents are highly proteolytic. They are actively pre-digesting the prey's cardiovascular architecture from the inside out, beginning the breakdown of tissue long before the snake actually swallows its meal. Elapids, like cobras and mambas, deploy a separate biological strategy using neurotoxins. Instead of degrading tissue, this pathway targets the neuromuscular junction. This is the exact site where nerve fibers communicate with muscle tissue across a microscopic gap known as the synaptic cleft. Here in the synaptic cleft, we see the first neurotoxic strategy. Mamba venom injects fasciculins to block the enzyme acetylcholinesterase. Normally, this enzyme clears acetylcholine transmitters. With it blocked, acetylcholine builds up, causing receptors to overfire and triggering severe muscle contractions known as tetany. Cobras execute the opposite strategy using alpha neurotoxins, a method relying on molecular mimicry. These alpha neurotoxins are shaped exactly like the acetylcholine molecule. This allows them to slot directly into the nicotinic receptors, physically jamming the connection so the real nerve signals cannot get through. The physiological result of this blockage is total communication failure, leading to immediate numbness, flaccid paralysis, and rapid failure of the diaphragm. While hemotoxins physically dismantle the body structure, neurotoxins act as an elegant override code. They bypass tissue damage entirely to directly shut down the prey's central operating system. All of this extreme biological specialization traces back approximately 170 million years to a single set of salivary proteins, an evolutionary origin known as the Toxicology hypothesis. Over millions of years, gene duplication combined with natural selection allowed these basic proteins to diversify into the highly specialized toxins we see today. This rapid mutation is driven by an ongoing biological arms race between snakes and their prey, a dynamic clearly visible in animals like the opossum. To survive snake strikes, opossums evolved silent substitutions in their VWF gene. This is the exact genetic sequence that codes for the hemostatic blood proteins targeted by the venom. In response to the opossum's resistance, the snakes underwent further gene duplication. This slightly altered the net charge and hydrophobicity of the venom molecules, allowing them to bypass the prey's new genetic defenses. Because of these evolutionary pressures, venom is highly dynamic. It functions as a biological software program, continuously patching and updating itself to overcome prey immunity. Because these venom proteins have evolved to target exact biological functions with extreme specificity, they offer an ideal template for human pharmacology. The strong analgesic properties found in specific neurotoxins are currently being reverse-engineered into powerful painkillers. These target pain receptors without triggering the severe side effects often associated with conventional opioids. Similarly, compounds that originally evolved to violently crash a prey's cardiovascular system have been isolated, synthesized, and deployed as modern ACE inhibitor medications to treat human hypertension. Developing these treatments requires the raw biological material itself, meaning researchers must manually extract liquid venom from live specimens. This extraction is also the foundation of serotherapy. Technicians inject carefully dosed, minute amounts of raw venom into host animals like horses or sheep, prompting their immune systems to produce the antibodies needed to create life-saving antivenom. The evolutionary pressures that forged this biological weapon have simultaneously provided the structural templates needed to synthesize highly specific medical treatments.